Okay, hello everyone, and thank you so much for coming to our class today. We are going to look into lesson number 3 for our biology grade 12. We are going to look into the chemicals for life. So it's going to be very interesting because we always use chemistry in order to, I would say, explain what's happening in the body. That's because, we are what we eat, and we are just, I cannot say just, but we are an assembly of molecules and ions. That's what you're going to see that with me today. So what you're going to learn today, we are going to describe the structure of important biochemical compounds, including carbohydrates. We're going to see what are those carbohydrates, and then proteins, lipids, nucleic acids, and explain their functions in the cell. Then we are going to look into the common functional groups within biological molecules. That's because all of these molecules, they need to interact. They have to just create some bonds to each other so that, for example, when we are going to have several amino acids, they must have a bond in between them so that we will be able to have a chain of amino acids that we call a protein. Or for example, we need to have several molecules of glucose so that we'll be able to have a molecule of glycogen. And in your body, we do have glucose molecules that will be used. We can also store them under the form of a glycogen and then in the liver. And then when we need more glucose, then the glycogen is going to provide us as a source, is going to provide us with enough glucose molecules. So as you see, these are all different types of chemical reactions that take place in the body. We also have enzymes that are in charge of course, facilitating that type of chemical reactions. And this is what we're going to look at, I would say, much more into details today together. Then we are looking into, again, main types of biochemical reactions. What is oxidation? What is reduction? Hydrolysis and condensation. All of them are very, very simple. Oxidation is when a molecule is losing electrons. So at the end, it may become positively charged or it may become neutral. On the other side, the reduction is when a molecule, after a chemical reaction, is gaining electrons. And those then at the end, the product may become negatively charged, or it may become neutral. And what is interesting is that in all of these different enzymatic reactions, we do have electrons that jump from one molecule to another molecule. And then we will see the jump that's a way to say that, of course, we will see the exchange, I would say, or the movement of electrons from one enzyme to another enzyme. And those movements of electrons are going to change the charge of the proteins, for example, and that change in the charge of the proteins may now end up provoking a change in the configuration of the protein, so that if the protein is an enzyme, for example, when this is going to be charged, this protein now can change its configuration, and then that enzyme may now show its catalytic site, for example, so we can say that an enzyme become active. So these are different ways that we can now change the structure or the configuration of proteins. And here I just gave you one example of enzymes. And again, for that change in the configuration or the shape of the protein or the enzyme, we need, of course, electrons. Then we are going to look into, for example, condensation. This is where we will see that in between two different molecules, when they are going to create a bond between themselves, we will see one OH and one H atom that are going to leave those structures in order to give us one molecule of water, so H2O. And on the other side, we will see a bond that will be formed. And this is what we call a condensation. We're going to look at it again in detail in a few pages. Then we are going to draw three-dimensional molecular models of important biochemical compounds, including carbohydrates, proteins, lipids, and nucleic acids. Why we are talking about different three-dimensional molecular models? That's because nothing is flat in nature. And you can start by looking into atoms around you. We do have atoms like hydrogen. They can share one electron with another hydrogen atom. They can also share one electron with another, I would say, group of atoms, for example. So here we will see that H or hydrogen can always create a bond with many types of molecules. Then we can look at, for example, carbon. Carbon can share up to 4 electrons with up to 4 different atoms or different groups of atoms. Here it's very interesting because now you see that when you have the possibility to have 4 different bonds between one atom and up to four other compounds or molecules, we will see how many possibilities we can have in order to have a spatial configuration. We can also talk about oxygen, where it can exchange up to 2 electrons with, again, 2 different molecules or atoms. Here we see that oxygen is able to give us, again, few configurations. And we can also look into ammonium or, again, nitrogen that can exchange up to 3 electrons with different atoms or molecules. And here again, it will give us lots of possibilities for having 3-dimensional structures in the space. And when I'm talking about different types of configuration, this is because, for example, if you're having, again, two hands, if they are flat in 2 dimensions, they may not interact with each other. However, if those 2 hands are now having some configuration in space, they are able to find somehow some way of interaction in between, of course, molecules and atoms that are making those hands. So this is why it's extremely important to understand the 3-dimensional structure of, again, DNA, of RNA, of proteins, carbohydrates, and also because in order for enzymes to recognize their substrates or the molecules that they usually transform or break down, those enzymes need to recognize a specific structure. And the structure is coming from the 3-dimensional structure of the molecules. So it's very important. I repeat again, we talked about hydrogen, about oxygen, about, again, nitrogen, and about carbon. Each of them is able to share up to again, hydrogen is 1 electron, oxygen is 2 electrons, so 2 bonds, nitrogen 3 electrons, so 3 bonds, and carbon can exchange up to 4 electrons, so it can have up to 4 bonds with different molecules or atoms. Now, let's look into the functional groups. We call them, usually you see them with the letter R, capital R. These are reactive clusters where much of the bonding takes place in biological molecules. So common functional groups in biomolecules are listed in the following page. I'm going to cover that now. So here you are going to see, again, in any natural molecule, you will see hydroxyl. So when we see hydroxyl, we are going to look into R, which is, again, that group of atoms, and OH, which is the functional group. It means that this is a group of atoms that are able to exchange. One atom can leave, like, for example, for OH here, hydrogen can leave the molecule of ROH, and then oxygen is now left with 1 electron, and it needs to have another electron in order to become to complete its last orbital. So the second bond of oxygen here is going to be available in order to interact with another molecule. So here, for example, we can have this type of, again, like ethanol, this molecule. So here R is here, this area, C2H5, and then we have OH. Now, the hydrogen can leave the molecule, and then the oxygen will be readily available in order to exchange its last electron with another compound. So this is how we are going to see, again, many chains that are happening, many chains are being produced. We are going to see chains of, for example, again, glucose that are going to be produced because of those functional groups. Now the second functional group is carboxyl. So here, as you see, we have an R, a radical, a group of atoms R. We have a carbon, and as you see, carbon has a one bond with the R, one bond with hydrogen, and two bonds with oxygen. So here we can now imagine that these two bonds between C and O, one of them can be opened, and then oxygen will be able to share one electron, so it can create one bond with another molecule or atom. So here we can talk, of course, the name of compounds are aldehydes and ketones. So the ketones and aldehydes, both of them, they do have a bond between carbon and oxygen. That's a double bond, of course, and that double bond can now be opened, and one of the bonds, so again representing two electrons, can now allow the functional group to bond or to fix the group that is already fixed to or bound to another molecule or atom. We also have carboxyls here, and we have now amino. For the amino, this is what we see mainly in of course, in the majority of cases, in proteins. So this is between amino acids that will be able to see this type of amino groups. We also have sulfhydryl, RSH. We call them thiols or thiols. So here again, on the right side, you're able to see that, for example, we can have a C2H5, then S, sulfur, and then the hydrogen can be gone with another, or to react with an OH, for example, hydroxyl, in order to make or to form one molecule of water, and the sulfur will be able now to bond again the C2H5 group into a molecule or another atom. We also have phosphate, very interesting and important. This is a group R, which is again bound to an oxygen, then to a phosphate. And here we will see that this phosphate is able to exchange one electron with the O minus that you can see on the right side, and then that O minus can now be linked or bound to another molecule. Here we can look into, for example, the structure of ATP or ADP, where we have adenosine, which is the R, the group R is adenosine, and then we can see one phosphate, we can call that adenosine monophosphate or AMP. We can have a second phosphate, so again, with a bond to the first phosphate, this molecule then becomes ADP or adenosine diphosphate. And we can also have up to maximum 3 phosphates in nature, of course, where we can call those molecules adenosine triphosphate. These are molecules that are made by mitochondrions, and they are bringing energy. How they can bring energy? That's because each time that you're having one of the bonds between two phosphates that is being broken, we are going to see the release of up to 8 joules of energy or heat. And because of that release of energy, ATP becomes ADP, because it will lose one phosphate, so it's not anymore triphosphate, but diphosphate or diphosphate. And then we will see the release of one phosphate, and then we will have ADP plus heat or some energy. And that energy now is being used for different types of enzymatic reactions in your cells. And of course, we do have thousands of different enzymes in our body as well, more than thousands, and all of them, they need ATP in order to again use the energy for doing their work. So their work can be breaking down, can be again catabolism, can be anabolism. These are different ways that we can see enzymes working. Now, looking into those again functional groups here, we can talk about methyl CH3, where carbon can still have one electron that can be shared with another molecule or another atom. We have hydroxyl, OH, we have carboxyl, we have amino, we have phosphate, carbonyl, and sulfhydryl or sulfhydryl. So these are, again, functional groups, and you will see only these functional groups in all of the enzymatic reactions in your body, in all of your cells. And then we are going to now look into the structure of each of the functional groups. First of all, let's talk about what is a redox reaction. A redox reaction is, again, oxidation and reduction. So redox, reduction, oxidation. So what they do is that they have a crucial role in our body's metabolism. So redox reactions are reactions that involve the transfer of electrons from one reactant to another. And when we are having the transfer of electrons, we may now be able to see molecules that are having more electrons to share with another compound. So it gives us the ability to, again, create new molecules, or it may be a loss of electrons that will end up giving us a neutral, for example, molecule. In this case, we can talk about the results or the results of a metabolic reaction. So here, what we can see is, for example, again, in this figure number 22 for the redox reaction, we can have a compound called A that becomes oxidized when A and B are going to interact. So A is, we call that a reducing agent. So this agent is going to become oxidized, and again, what happens is that it's going to lose electrons. So now that, again, A may become, of course, positively charged because it doesn't have, again, it's losing electrons. On the other side, and the opposite, is when we are having an oxidizing agent like B, it can be any molecule or any atom that is going to interact with the reducing agent, which is A. And then at the end of the reaction between A and B, we are going to see the exchange of electrons between A and B. So A is losing electrons and B is gaining electrons. Now you may tell me, okay, good to know, I understand, but why we are talking about electrons here? As I told you, my friends, these electrons that you can see in the reduced compound B, this can be glucose, this can be mannose, this can be any fat, it can be any carbohydrate, it can be any molecule in your body that can become reduced. And then as a product of a reaction, this is going to have extra electrons. Now imagine that that carbohydrate, for example, that compound is now having two more electrons to share with a structure. Those structures are usually, not always, but usually enzymes. And those enzymes are made of amino acids. And those amino acids are giving a shape to the enzyme. However, if the enzyme is not activated or active, it will not do anything because its configuration or its form is in an inactive form. When an enzyme is going to be in the proximity of a reduced compound like B here, the electrons that are now available on compound B are going to be transferred to the structure of the enzyme. Now those electrons are going to jump between different amino acids that are making that enzyme. And now what happens at the end is that the configuration or the form of the enzyme is going to be changed in a way that we will see its catalytic area or section is going to be revealed because of the change in the structure of the protein due to the again transfer of electrons. And then we will see the transformation of molecule A. Now imagine, for example, that we are in your mitochondrions, and on the membrane, inner membrane of the mitochondrion, we are having a cascade of enzymatic reactions, and we have, for example, 6 enzymes within the inner membrane. Now we may have a substrate that will come, and it will be now the substrate of the first enzyme. It will now change that substrate A into the substrate B. Now that substrate B is going to be recognized by the second enzyme that will then transform B into C. So now we will see a cascade of transformation of a compound A that becomes B, C, D, E, and then at the very end, we are going to see another molecule, because while during the cascade, we are going to do a transformation, we can now add a phosphate, we can remove a phosphate. Now imagine that an adenosine can now become AMP, ADP, ATP, adenosine triphosphate. We can now see, for example, that a molecule of glucose is becoming glyceride, and then it's now going to become different types of molecules until the time that we will see all of the cascade, the enzymatic cascades are going to allow us to make from one molecule of glucose to transform it to different molecules. And then at the end of that cascade, enzymatic cascade, we will be able to produce ATP, or adenosine triphosphate. This is why we need to understand those oxido or redox or oxido reduction reactions in the body. And it's very interesting, because then how we can use the information by making new drugs, by understanding how toxins are working, by even sometimes using toxins at a very low dose in order to just do some experimentation in cells in order to test new drugs. So this is why we need to understand how our body works. And this is just a very simplified explanation of what is the redox reactions. Now, talking about biological macromolecules, so these are molecules that are big. We do have different classes or groups of macromolecules. We have carbohydrates. These are oligosaccharides and polysaccharides. What does it mean? Saccharide is a sugar, and polysaccharide means a macromolecule where you don't have only one saccharide. You have the same identical saccharide that is now making a chain. We call that a polysaccharide. We can also have oligosaccharide, means that you do have different saccharides that are now being in a chain. And here we can again talk about simple sugars like glucose, like fructose, which is the sugar that you can find in fruits. We can also then, for another class of macromolecules, we can talk about the fats and lipids. Here we have di and triglycerides, and the specific example are phospholipids. These are molecules that are making the cell membrane. All of the cell membranes that we have in our body, they are made of these units of phospholipids. And of course, the subunit are glycerol and fatty acid that we're going to see in a few minutes. And examples, we can talk about oleic acid in vegetable oils and glycerol. Now, another class of or group of macromolecules are proteins. So we call them proteins, and examples of proteins can be enzymes. For example, there's just one. Their subunit are amino acids. It means that we need to have several amino acids to be bound to each other in order to give us a chain of an amino acid. Now that chain, we call it a protein. Now the protein can stay in a line. Okay, this is again a primary structure. It can now have some structure that we call sheets or helices, beta sheets or alpha helices. So here this is a secondary structure. They can have a tertiary structure, so a three-dimensional structure, and they can have a quaternary structure where several tertiary structures of protein, they are all assembled together, and they will give us the quaternary structure of a protein. And here we can give you an example of heme, which is a molecule that will allow you in your red blood cells to grab and to fix oxygen, and then to release it in areas in your blood where we don't have enough oxygen and the pressure of the oxygen is not high. This is where the oxygen is going to be released. Now, hemes are molecules you have in your red blood cells, more than 100, 200 hemes. These are molecules of hemes. They can all, again, fix or grab oxygen coming from your lungs, and then they will be circulating with the blood, and they are going to find themselves in areas, in capillaries, where the pressure of oxygen is very low. So here they are going to release oxygen. So as you see, these are the uses of different types of proteins in the body. Another class of or group of macromolecules are nucleic acids. So here we can give the example of DNA and RNA. So the specific examples again are already there. We have mRNA or messenger RNA. We have tRNA or transfer RNA. And the monomers for them are nucleotides that we're going to see as well. And the examples are phosphate groups, which is with plus sugar, plus one of the 5 nitrogenous bases. These are puric and pyrimidic bases. We don't go over that today. Of course, we'll go over this when we are going to look into the genetics. Then we have to look into other types of chemical reactions. This one is the condensation. So what is the condensation? This is the dehydration synthesis. It means that when you're having two molecules interacting with each other, in order for them to make a bond and to stay more stable, they are going to lose water. This is why we call that the condensation or dehydration synthesis. We also have the hydrolysis reaction, where one molecule of water is going to be used in order to give us a hydroxyl or OH and a hydrogen, and then that HOH or H2O is going to allow two molecules to separate. And when they separate, one again, if they need, of course, they are going to have one electron to exchange and to share in order to become stable. So here we're going to see OH. So hydroxyl is going to again bond to one molecule and hydrogen to another molecule. And now we are going to have two molecules that are going to be stable. So here, the example of condensation is in between, again, for example, alpha D-glucose and D-fructose. So here on the left, you see the glucose, and here you see the fructose. You see fructose has you see 1, 2, 3, 4, five carbons. Glucose has, again, 6 carbons. So here we see that when we see the HO2 that will be eliminated, so you see that H here and HO here, we will see the formation of a bond in between oxygen of the glucose and here the carbon number 4 of the fructose. And then at the end, we will be able to see, again, sucrose. So sucrose is made of one molecule of glucose and one molecule of fructose. For glucose, glucose has 1, 2, 3, 4, 5, 6, again, carbons, and the fructose has 1, 2, 3, 4, 5 carbons. So now both of them, they are going to, under a condensation reaction, and they are going to give us one molecule of water plus one molecule of sucrose. And of course, when you are eating again sucrose is going to again undergo some changes, undergo some transformation in the body, so that we'll be able to again break down that alpha glycosidic link, so that we will now have one molecule of fructose and one molecule of glucose, so that our body will be able to break down and glucose and fructose in order to produce ATP. So the glucose and fructose, of course, they're both going to undergo an enzymatic reaction, the cascade of enzymatic reactions in mitochondrions in your body, in your cells. And those mitochondrions are going to use glucose and fructose in order to produce ATP in the mitochondrion, that will then be able to be exported from mitochondrions and to be readily available as an energy, a source of energy, to different enzymes in your cells. Then we are going to look into carbohydrates. Carbohydrates contain a carbon, a hydrogen, and oxygen. And the ratio is always 1 to 1. It means you have always one carbon, two hydrogen, and one oxygen. So here we can see, for example, CH2O. And when you see CH2O between parentheses and N means that you can have CH2O, CHO. You can now have a chain of the same CH2O, or I would say carbon, hydrogen, and oxygen. Then we can talk about, of course, we'll go over that in the a few minutes. First, we can talk about monosaccharides or monosaccharides. What are those? These are one molecules of sugar. So here we can talk about, again, glucose, mannose, fructose, again, different types of sugars. So here mono means, of course, single, and saccharide or saccharide from saccharum means sugar. Now here, we can now giving you again an example of again a sugar. So here we are talking about glucose, and what you can see is that glucose is in a circular form. So what happens here is that you do have a carbon, the last one here on the top, that you can see on the circular, I would say the cyclic, I would say, structure of glucose that is shown at the bottom of this page, where you have a carbon, hydrogen marked in blue, and hydroxyl marked in orange. So what does it mean? It means that if you have a carbon within the cycle, that carbon is flat. However, when you're having a chiral carbon, which is the last carbon of, sorry, the carbon number one of this glucose, this structure, we will be able to see two different possible configurations. We can see on the left alpha configuration, where the OH or hydroxyl radical or group is situated underneath carbon number one, or we can have beta glucose, where the on carbon number one, which is the chiral carbon, we do have again the OH. So what does it mean? It means that now when we are having enzymes, in order to use glucose, those enzymes are having a preference in order to use more the alpha glucose. So in order to for us to break down beta glucose, we will need to undergo some conversion or transformation of the beta glucose and to make it alpha, and then to use it. This is why when we are using one type of the glucose, because of its configuration, we may not be able to necessarily obtain the same number of ATP, or those energy source of energy molecules, in the cell. So at the end of the day, why we need to understand that? Because we need to understand how, again, organs and organelles are working in our body. Now, underneath here, you can look into the structure of the glucose. As I told you, this is in 3 dimension. It's not a flat molecule. So we have alpha glucose and beta glucose because of the possibilities that we have when we are having a chiral or chiral carbon in a molecule. Then we are going to look into polysaccharides or polysaccharides. It's a complex carbohydrate composed of 100 to several thousand monosaccharides, subunits joined by glycosidic links. For example, starch and glycogen used for energy, and cellulose and chitin used for structural support. Of course, chitin is the protein that we see in the exoskeleton of insects. Cellulose is also the structure or the molecule that we see mainly in plants. And now, for glycogen, again, as I told you, this is just a set of several molecules of glucose, and it can be stored in the liver. I mean, you are going to have glucose molecules. They're all going to be absorbed by the liver cells, and they're all going to be bound to each other in order to give us a chain of glucose molecules that we call glycogen. Now, this is going to be stored, because when glucose is stored, it cannot be broken down or used by mitochondrions. It needs, a mitochondrion needs to have molecules, single molecules of glucose. So when we have a chain of glucose, we will be able to store it without using it. So we will always have a type of glucogenesis and gluconeogenesis. It means, sorry, glucolysis or glucolysis and gluconeogenesis. It means that we always have making up glycogen, and of course we can have again reactions that will allow us to break down glycogen into glucose, so that our mitochondria will be able to use those glucose for making again energy. So the same thing for the cellulose applies here. This is for the plants, and of course the same thing applies for chitin, which is again it's making up the exoskeleton of insects and crustaceans. Perfect. So now here we do have some examples of those poly or oligosaccharides. So here on the top we have, for example, glycogen, and here we do have starch. So these are all different molecules of sugars or saccharides, and they're all linked or bound to each other. So on the left you see the again it's, I would say, type of you can see in bigger again images what is the structure of the glycogen, starch, and of course cellulose in plants. Perfect. Then let's now talk about fat and again and fatty acids. Glycerol is a 3-carbon alcohol containing a hydroxyl group attached to each carbon. Glycerol or glycerol forms the backbone of triglyceride molecules. Triglyceride or triglycerides. So as you see, I'm always using the same way of reading those names, because one of the way that we read is the English form, and one of them is the American form. So we can have triglycerides or we can have triglycerides. Both ways are okay for you. So you can pronounce them easily. There is no issue with that. Now, after glycerols, we can also talk about fatty acids. What are those fatty acids? They are long hydrocarbon chains with a single carboxyl group, COH, and at one end. Fatty acids can be saturated, monosaturated, or polyunsaturated. We're going to see what is that. Saturated fatty acids have no double bond. So we always have single bonds between carbons. These are saturated fatty acids. Then we have mono unsaturated. They have only one double bond, and polyunsaturated fatty acid have 2 or more double bonds between carbons. Animal fats such as butter and lard, this is coming from the pig, are composed of or triglycerides with mostly saturated fatty acids. Saturated means that all of the bonds are simple bonds between carbons. Chains and are solid at room temperature. Plant oils contain mono and polyunsaturated fatty acids and are liquid at room temperature. Perfect. So here we do have different again types of fatty acids. On the left you see the saturated fatty acid, and when you look into the carbons, you can see that the bonds between all of the carbons are simple bonds. So this is a saturated fatty acid. Here for the monounsaturated, we see that there's only one double bond between 2 carbons. Why we talk about the saturated and mono or polyunsaturated again fatty acids? That's because there are differences in the use of these fat molecules in the body. For example, saturated, first of all, for the level of energetics in the mitochondrions, the amount of the energy that is produced based on the breakdown of these fatty acids is different, slightly different, but they are different. On the other side, few of these types of saturated or monounsaturated fatty acids, they can also trigger some physiological reactions. And here, I will not go again over that discussion today, but they can now cause different types of disorders if they are used at a high concentration or over years. I'm talking about different diseases or progress of different disease conditions in the body. Now, saturated versus unsaturated fatty acids. Here we can talk about triglycerides, so made from the monomers glycerol joined with 3 fatty acids. The bonds between the hydroxyl and the glycerol and the carboxyl of the fatty acids are called ester linkage. So here you can see the triglycerides here, and you have these 3 fatty acids. This is a triglyceride. Perfect. So now, again, of course, here as well. Now, phospholipids. Phospholipids are made of phosphates and lipids. And when you see here the lipid bilayer, you see the structure of the phospholipid. One layer, outer layer, is made of a chain, I would say, is a full layer of phospholipids, and the inner layer is also made of a layer of phospholipids. We call that a bilayer for the membrane. That's because the polar or hydrophilic head is always situated where we have fluid or water, and the polar hydrophobic side is going to be in the side where again it should be another hydrophobic side. This is why when we have phospholipids, the outer or the polar head is situated where the outside of the cell, and the other side is faced to the inner side of the cell. And in the middle, you see how the polar and non-polar, I would say, structures are faced to each other. And then, of course, we can talk about proteins. I already talked about those. For the proteins, we always have an amino group, and then we have a group R and carboxyl group here. Now we have 20 amino acids that in the nature we have more, but in our body we do have 20 amino acids that we use in order to make our proteins and different types of enzymes like valine, leucine, isoleucine, methionine, phenylalanine, what else, aspartic acid, glutamine, histidine, etc., etc. Some of these amino acids are polar, meaning that they can change their charge, and if they can change their charge, they can now also change their configuration. So if you're having several polar amino acids in a protein, that protein, once it is again being in an environment where the pH changes can change, and the charge, overall charge of the protein or the enzyme can be changed, then we will be able to see a change in the configuration of that protein. And if this is an enzyme, it will then be able to have some catalytic sites or regulatory sites, and then those sites are going to be shown when the structure of the protein becomes charged. And of course, if now we remove the charge and the protein becomes neutral, for example, then we will see that the protein or the enzyme is not active anymore. Okay, so this is for this. That was for talking about this one. Now, here we are talking about the again a polypeptide. Polypeptides are made of several again, proteins, and here we can talk about different again the structure which is tertiary. This is when we have the one chain of amino acid, as I already mentioned, that is going to create a three-dimensional structure, and then we are going to see different ionic bonds. You see disulfide bridge. We can have hydrogen bonds, we can have protein kink, and then we are going to see several types of enzymes or again proteins with a tertiary structure that are going to have different shapes, and they're of course going to have different functions in your body. So here, of course, we can talk about the primary structure at the top. It's just a chain. The chain can become again a helix. We call them alpha helix. They can be pleated sheets. We call them beta sheets. And of course, they can give us a tertiary a molecule or protein, and then they can give us a quaternary structure. Of course, please read the book chapter on this course as well. Now, we can also have protein denaturation. It means that when you are heating up a protein, you are going to affect the bonds in between those amino acids. You may break them. We may even now change again, the configuration of the protein, and then the protein may not come back to its original configuration. In this case, the protein is, we cannot call that a dead protein, but it becomes denatured. So it loses its original nature. And then what happens is that in a body, for example, in a cell, if we have denatured proteins, it may end up killing the cell. But if the cell survives, the cell is going to reproduce the original protein, and they're going to replace the denatured protein so that the cell is going to survive. So the survival rate of the cell when exposed to a heat is, again, it depends on, again, the severity or degree of severity of the damage to those cells and to those enzymes. But when an enzyme is denatured because of fever, for example, it can be a fever as well. In this case, if the cells are not dead, the body is going to compensate by creating new molecules for the enzymes. So my friends, I am done with, of course, this lecture. For nucleic acids, you can, of course, read that on your own. This is, again, just an introduction. Please read the book chapter, of course, for this class. And of course, we are going to, again, I'm going to see you next week for the next class.